synthetic theory of evolution, the process upon which synthetic theory of evolution is based on, the most important one is the mutation. Okay, now what is mutation? Mutations are any change in the genotype. It can be, in a wide sense, it can be referred as a mutation. Mutations are changes in the genetic material, either in the chemical structure of genes or in the structure or number of chromosomes. Okay, when it is in the chemical structure of genes, the change is referred as a gene mutation. When it is in the structure or number of chromosomes, we refer it as a chromosomal mutations. Okay, so any sudden heritable change in the genotype is what is referred as mutation. When a gene changes, the character it controls also changes, and these changed characteristics are referred as the variations, and these variations are inherited to the next generation. Okay, however, mutations provide heritable variations resulting from changes in the structure or number of chromosomes and the genes. Okay, so these are heritable, and it happens since it is in chromosome level or genetic level. Obviously this gets passed on to the next generation. Okay, now mutations can be classified into two major categories, right? The gene mutations and chromosomal mutations, that is where it is where the change is happening. Okay, so gene mutations are otherwise referred as point mutations, and these are caused by chemical alterations of individual genes. Right. Chromosomal mutations are otherwise referred as aberrations. Okay. Chromosomal aberrations or simply the aberrations there. They are caused by changes in the structure of individual chromosomes or changes in the number of chromosomes. Okay. Both are referred as chromosomal aberrations. Now we will look into in detail the gene mutations. Okay. A gene mutation is any change in the chemical organization of the gene that is replicated and passed on to successive generations. It is also referred as point mutations because this change happens in a single gene and it affects only a particular locus in a chromosome. I hope you remember what is a locus. Okay, so locus or the point in a chromosome, and since this change happens only in a single locus, it is referred as a point mutation. Okay, now genes basically, chemically they are composed of DNA molecules. DNA contains coded information in the form of base sequences, especially the base pairs. Since it is DNA, it is the information it is coded in the form of base pair sequences. Okay, I hope you remember what is a base pair. It is like C, T, G, C, A. I'm sorry, what do you call it? A, T, G, C. Those are the base pairs, and in a what you call different combinations of these sequences will be present in different genes. So a gene mutation takes place by rearrangement, addition or deletion of these bases in the DNA. Okay, so what all changes can happen in a gene mutation? There can be rearrangement. Okay, if it is, just imagine, for example, we have a sequence like right, A, okay, complementary T. There's a DNA, isn't it? So there will be double. Just imagine another A over here, then T, okay, a T, next T, A, okay, then G, C, right? Just imagine this is the base sequence of a gene. Obviously, the gene cannot be so small, but still, rearrangement in the sense, instead of here A, T, A, T, T, A, it could be A, T, T, A, A, T. Okay, getting rearranged. Okay, so that kind of a change is known as rearrangement. Okay, then it could be 1 minute. Okay, it could be additions. Okay, there can be a new base sequence introduced in the already what you call existing base sequences. A new base pair may be included. Okay, deletions, already existing base pair may be removed. Okay, so all these can bring about changes in the normal gene sequence, and that is what brings about the gene mutation. So any change in the order of these bases will alter the coded message that the particular gene contains, and as a result a modification of polypeptide gene. I hope you remember that whatever base sequence is coded there on the DNA, it will be transcribed to mRNA, and from there it is getting translated to amino acid sequence, isn't it? So whenever there happens a change in the base pair sequence, there will be change in the transcribed mRNA, there will be change in the amino acid codes, isn't it? So whenever there is a change in the amino acid sequence, the polypeptide or the protein formed from the amino acid sequence will also be modified, right? So as a result of this modification of the polypeptide chain produced, obviously it will result in the proteins, whatever role it can take up. It can, the proteins can be an enzyme, it can be other proteins, or it could be involved in the suppression of some enzymes or proteins, right? So the protein which is formed from the modified base sequence or modified DNA, it can result in modified enzymes or modified proteins. Just imagine if it is a modified enzyme. Okay, we can call it as a mutant enzyme. Okay, this may ultimately express itself by affecting a change in the physiology or development of the organism. You may have studied glycolytic pathway, you may have studied Krebs cycle, etc. All these are like multi-step process, and each step is controlled by an enzyme. So if an enzyme undergoes modification because of gene mutations, these metabolic pathways and physiological processes, all these can get like affected. Okay, so these changes are now considered, so these are the changes that can be considered as a molecular basis for evolution. Okay, so I hope it is clear, right? That is how gene mutations can become can result in what you call the evolution. So now what are the characteristic features of gene mutations? Okay, the most important feature of gene mutations are listed, like first one is genes are relatively stable. So what happens is there are less proportion of mutations happening in a normal condition. For example, in Drosophila, only about one gene mutates out of 20 gametes produced. So among the 20 gametes produced, there can be only a single gene mutation. Okay, so it is the proportion is very less. So since each gamete contains about 20,000 genes, you can imagine 20 gametes means it is actually 4 lakh genes. Among that, there is only a single gene mutation that can happen. So it is a relatively very low rate. So genes are relatively stable. Okay, it's not gene mutations, genes are relatively stable. Now secondly, some genes, they can mutate to several different allelic forms, and this is what is referred as the multiple alleles. Okay, I hope you remember the blood grouping of humans, isn't it? So single gene can mutate to form different forms, and that is known as a multiple allelism, right? Then gene mutations are the ultimate source of heritable variations. Okay, heritable changes or heritable variations. So gene mutations are considered to be the ultimate source of heritable variations in organisms. Fine. And most mutations happening, they are either harmful or it could be called as deleterious, and some are even lethal. That is lethal in the sense it can even cause death of the organism, right? And sometimes a mutant gene changes to become the original. Okay, it can change back to its original form, and this is what is referred as the reverse mutations. Okay, or it is referred as a back mutation. Very rarely that kind of a change can happen, right? Then we have is the like rate of mutations can be modified externally, experimentally. So there are agents that can produce mutations, and those kind of mutations are known as induced mutations. Okay, induced mutations are those mutations which are introduced by way of external agents, right? For example, X-rays, ultraviolet rays, high temperature, And then variety of chemicals can induce mutations, gene mutations. So, okay, then another characteristic feature is that mutation is the result of slight change in the chemical structure of DNA segments, and these DNA segments are what is referred as a gene. Okay, so the gene mutation result from slight change in the chemical structure of a gene. Okay, and these gene mutations are random in the sense that the environmental influence that activates the chemical change cannot induce a particular mutation. That is, it needn't be specific. Okay, so whenever mutation is induced in a DNA, it can be one or the other gene. It needn't be very specific. Okay, and it occurs without any regard to the usefulness or the need of the gene to the organism. Okay, and another important feature is that they are spontaneous. Okay, and are what you call undirected by the environment. So it can be in any way, and it is spontaneous. It is random and they are spontaneous. Okay, they are spontaneous, and mutations are thus accidental, undirected, random, and like chance events, right? Then some mutated genes may be pleiotropic. Pleiotropic in the sense mutations, some mutated genes may be pleiotropic in the sense it can affect more than one character. For example, the allele that produces vestigial wings in Drosophila also causes a modification in the balance organs. Okay, so in gamete production, so they affect many characters at the same time. So those kind of genes are known as pleiotropic genes, and most gene mutations are recessive. Dominant mutations do not occur. What do you call? Rarely occur, but most of them are recessive in nature. Okay, so they can be detected at once, while recessive gene mutations can be expressed only if a homozygotic recessive is born. Okay, homozygote in recessive character is born. Okay, that is it goes actually undetected. That is it, right? And most mutations, they occur at any stage in the development of an organism. They may be somatic or germinal. It could be in reproductive cells or in vegetative cells. And mutations are universal. That is occurring in diverse organisms from viruses to human. Okay, uh, so any kind of organisms can like experience genetic mutation or gene mutations, and mutations are relatively persistent, though it is not permanent, but it is relatively persistent. That is, a mutant gene may be transmitted through many generations without undergoing any further change. However, after a number of generations, it may mutate again, or it may revert back to its original condition. That is, it can take up reverse mutation. Okay, so it is persistent, but it is not permanent. I hope you got the difference. Okay, so genetic mutations, the main characteristic features are genes are relatively stable, and the genes can mutate to form multiple alleles. The ultimate source of heritable variations are genetic mutations, and most mutations are harmful or deleterious, and some are even lethal. Then there can be external agents that can bring about induced mutations. Some of the gene mutations that they can like revert back to its original condition, and the gene mutations result from slight changes in the chemical structure of DNA segments. The mutations usually they are random and spontaneous, even undirected, and like accidental. Then the mutated genes can be pleiotropic, and most of them are recessive, even though dominant mutations can occur in dominant, like mutations can be dominant, but mostly the gene mutations are recessive, and most of them go undetected. Okay, they are universal, and they can occur at any stage in the development of an organism, right? So these are the characteristic features of gene mutations. Okay, chromosomal mutations we'll be looking in detail in the next presentation. Thank you.